So I was just reading about oncogenes, how to write it as a short note. Basically, it is a mutated, so we put the heading as definition. When we see a normal cellular proto-oncogene, but it gets mutated or overexpressed, that forms the oncogene. That you draw 4 arrows and we can say that it promotes cell proliferation, survival and malignant transformation. There is gain of function mutation in these genes that cause it to act in this manner, dominant manner. In oncogenic activity, usually even a single allele mutation alteration can also cause such mechanisms. So we just draw a small table where we depict proto-oncogene on the left side, right side oncogene. So left side we will write proto-oncogene is a normal gene, right side mutated gene. Left side it regulates normal cell growth versus uncontrolled cell growth, right side. Again, proto-oncogene responds to growth signals. Yes, oncogene, it will function independently on its own. The regulation mechanisms are disturbed. Proto-oncogene is physiological, oncogene is pathological. So a small table to differentiate between this. And you just write functions of proto-oncogenes. Put arrows everywhere, put a circle and surrounding it, we can write it. No? Instead of writing it as a paragraph, I will write it as growth factor receptors, intracellular signal transduction, transcription factors, cell cycle regulators, anti-apoptotic proteins, and they regulate normal cell proliferation. All this can be mentioned as functions of proto-oncogenes. So we define, then we put a tabular column, then we write functions of proto-oncogenes. Then you come mechanism of oncogene activation. This is important. So we have a gene that gets genetically altered. So the oncoprotein activity increases and then there causes autonomic cell proliferation, which was normally supposed to be regulated. Here it becomes automatic. And that proliferation leads to your malignant transformation. Uncontrolled proliferation of cells, which is completely abnormal and not necessary for the body. The mechanisms that cause this: point mutation, 1. Gene amplification, 2. Chromosomal translocation, 3. Retroviral insertion. So these are the 4 major mechanisms that alter the. So point mechanisms also. It produces just a single point in the gene itself. So usually it is seen with the RAS gene, KRAS, HRAS, KRAS with pancreas, HRAS with colon. Then there is gene amplification. This increases in gene copy number. That leads to increased protein production, like HER2 amplification, EGFR amplification, N-MYC amplification in neuroblastoma, L-MYC amplification in SCLC, EGFR amplification is pharmaceutical carcinoma. All this is gene amplification. Translocation, T922 BCR-ABL in CML, 1418 T1418 BCL2 follicular lymphoma. Burkitt lymphoma is also as well. T814 I think. These are reasons where it creates a fusion protein which is not necessary but leads to a strong promoter sequence that leads to uncontrolled proliferation of cells. Then retroviral insertion by viral promoters causes overexpression and it integrates into the proto-oncogene causing its overexpression. It has been explained with animal tumors, and that is how the initial oncogenes are actually like I think the example would be the Rouse sarcoma, RSV virus, HPV, this can come under retroviral. So once this is happening, what are the biological effects? Autonomous cell proliferation, loss of dependence on growth factors, increased cell survival, resistance to apoptosis, cellular immortalization, This in turn, everything together, we put a bracket and tell tumor progression, metastasis, or invasion and metastasis of cancer. So just to recollect, we initially defined what is an oncogene. We put a small table to differentiate proto-oncogenes versus oncogenes. We put normal functions of proto-oncogene. Then we write a small flow chart on mechanisms of oncogene activation. Then we write the 4 major mechanisms with its examples. Then we write the biological effects of oncogene activation. Then we say clinical significance. See, HER2 amplification, BCR-ABL fusion, and MYC translocation, they are all oncogenic activators, oncogenes which are being activated. So this is a diagnostic marker for certain cases. HER2 is for breast, MYC is for Burkitt lymphoma, BCR-ABL we all know is for CML. This is a diagnostic marker. Prognostic marker, prognosis, N-MYC amplification, neuroblastoma, poor prognosis. HER2 amplification, we know HER2 is, even though it is targetable, we know HER2 breast cancers are kind of aggressive. So that acts as a prognostic marker. Then therapeutic targets. BCR-ABL fusion can be targeted with imatinib, dasatinib. HER2 can be targeted with trastuzumab. EGFR, we have osimertinib, geftinib. BRAF, again we have dabrafenib, vemurafenib. Yes. So these are all genes, oncogenes basically, which are targeted. This is the clinical significance, diagnostic, prognostic, therapeutic. And one more thing is that oncogene and tumor suppressor genes may be similar in a way that they are all dysregulated. The only thing is that oncogene is usually due to gain of function, whereas tumor suppressor gene is due to loss of function. It needs to suppress the tumor, but it loses its function. Oncogene, proto-oncogene normally regulates. Oncogene gains the extra function, unnecessary function, and creates cell growth, so stimulates growth. Tumor suppressor gene will inhibit growth. Genetics is usually maybe recessive in tumor suppressor, but it is dominant in oncogenes. They say that in previous also I had mentioned that we require at least one allele to be altered in a gene to cause effect in oncogene. But but in tumor suppressor, we need both the alleles to be lost or mutated or targeted. This is how I think I will write an answer for the question oncogenes as a short note.